I always break that down into two separate aspects, the generate and then the analysis tools. From the generate aspect, I think the use of microwell technology such as corneal neoplasia has really changed the game, at least for our customers. And that's because it's become very simple to generate large number of very uniform spheroids. If you look at some of the other methods before, when you had to do just low attachment plates, you had real issues. We call them megaspheroids, that you get this big glump after a while. So the ability to control size and to work with large number of spheroids with these microwell technologies has been a game changer. In terms of analysis tools, you're seeing great advances in imaging technology, different types of spinning disc confocal, so you're really getting a picture deep within the organoids. And again, I'd be remiss not to mention large particle flow cytometry like BennoBio has developed, where you can look at large number of intact 3D particles very simply.
The tools that I found most effective recently are the ultra-low attachment services and the ability to utilize 3D models more homogeneously, cost-effectively, rapidly. Not that 3D models weren't out there before, they certainly were. Hanging drop methods and others were certainly suitable and set the stage for what we do today with 3D models. But now we have a democratized system where it's really cost-effective, it's homogeneous, and sort of ubiquitous worldwide to be able to do these fast and cost-effectively for all of the types of studies you need to do, immuno-oncology, small molecule discovery, biologics, etc.
For us, the biggest tools that we have utilized are the corneal neoplasia plates for being able to efficiently and reproducibly scale up the spheroid production for our preclinical use. We are hoping to further develop this relationship with Corning in developing higher throughput methodologies for manufacturing scale spheroids for our clinical use.
I think the organoids have a lot of strengths when you're studying human genetics and genetic mutations. They're often unique to the human species. In fact, there's entire genes that are essentially unique to the human species that can cause disease, like apolipoprotein L1, which is a major risk factor for kidney disease. It really, the mutants that cause disease are unique to humans, and the gene as a whole is unique to non-human primates and humans. So, we've got these specialized opportunities where we want to now develop gene therapies, for instance, and the target of those therapeutics is the human genome. And the only cells that have human genome are human cells. And we can use the organoids as the best approximation in those situations for what's going to happen in people.
Yeah, so the tools that I find most helpful for working with 3D models, organoid models in my lab are using different low attachment products like Corning ULA plates or spheroid plates, RL plasia plates for generating 3D models. But certainly if I'm looking for something more complex, right now hydrogels like Corning Matrigel Matrix are really essential for generating and maintaining these complex organoids.
Working with an ECM like Matrigel is definitely going to be the easiest solution if you're trying to generate functional organoid quickly. It's very difficult to get to move away from ECMs, especially complex ECMs like Matrigel. So I was a huge fan of working with it for cortical and brain organoids.